Assalamualaikum students. So today we will start the histology of the gastrointestinal system. First of all, the gastrointestinal system is that system that helps to ingest the food, then break down it, then digest and absorb the nutrients while eliminating the waste products from the body. Now this gastrointestinal consists of the tubular passage in which the food is processed and broken down, digested and absorbed, and then is eliminated. So the organs associated with this system are the number one, this system starts from the mouth, which opens up into the oral cavity. Oral cavity continues in the funnel shaped structure, which is the pharynx tubular structure. Then this tubular structure continues in the esophagus tubular organ, and then esophagus continues into a dilated part of the digestive tract, that is the stomach. Then it comes to the again a narrow part, which is the small intestine. Then this small intestine is continuous with somewhat wider part, which is the large intestine, which ends at the anus. Now, as you know, the function of the GI system is to digest the food. So this system is also related with some glands. Salivary glands, they produce saliva, which cause emulsification of the food, and the enzyme present in it starts digesting the carbohydrates. Then the liver, which is the major organ of the body, major gland of the body, and the pancreas and the gallbladder. So gastrointestinal tract is associated with four types of glands: salivary glands, liver, pancreas. Gallbladder actually stores the bile, but it has got function of secretion also. So now we will study the general histological structure of the GI tract. So as you know, GI tract consists of a wall. As it is a hollow organ, it has got a wall. This is the wall of the hollow organ, and this is the empty space in between, which is known as the lumen. So this is the lumen. Now this wall of the hollow organ of GI tract, whether it is esophagus, whether it is pharynx or stomach or small intestine or large intestine, it consists of four layers. First layer is the mucosa. Second layer is muscularis submucosa. Second layer is the submucosa. Third layer is the muscularis externa. This is the third layer, this is the second layer, and this one is the first layer. Now the last layer, which is not shown here, is the Sirosa or adventitia. Now the mucosa. So mucosa is the innermost layer. So this is the innermost coat, means it is facing the lumen. The layer facing the lumen is the mucosa. Now as you know from your previous learning in the first year, that all the luminal surface of the body, they are lined by the epithelium. So the mucosa, it is the most innermost layer, and in this mucosa the most innermost layer lining facing the lumen is the epithelium. So the epithelium is always associated with lamina propria, means epithelium, it is always associated, you know from your knowledge of first year, that epithelium is always associated with the loose connective tissue. Loose connective tissue. So this is the This is known as the lamina propria. This loose connective tissue is known as the lamina propria. Now, in the tubular structure of GI tract, another layer is present in the mucosa, the third layer, which is the Muscularis mucosae. So, it is present only in the lining of the tubular structure of the GI tract. Now, the epithelium. As you know, the epithelium lines the luminal surface of the hollow organs of the body, and epithelium lines the surface of the mucosa. So, it varies in different parts of the alimentary canal because of the specific function performed by each part of the digestive tract. You know it very well. When you need some protection, so there is stratified epithelium. But when we need to secrete something or to absorb something, so the epithelium changes to simple, that is the simple columnar or simple cuboidal. Also, this epithelium invaginate to form the exocrine glands in the wall of GI tract. So, epithelium, it varies from region to region. We have understood it. In the esophagus and upper part of the anal canal, we have got stratified squamous, non-keratinized. Also in the oral cavity and the pharynx, esophagus and lower part of anal canal, the epithelium is stratified squamous, non-keratinized. Why? Because the food particles are still enlarged in the oral cavity, in the pharynx and in the esophagus, and we need to protect our lining from this hard food particles, which are still large in number, large in size, and in the lower part of anal canal there are hard feces, which all the nutrients have been absorbed from the in the GIT, and the waste product becomes hard due to absorption of water also. So, in the lower part of the anal canal also stratified squamous non-keratinized epithelium is present. Now, in the stomach the epithelium changes from simple columnar to simple columnar, from the stratified squamous non-keratinized epithelium to simple columnar epithelium, because here this simple columnar epithelium has to secrete and it has to absorb certain substances. In the small intestine, we have also got simple columnar epithelium. So, it is absorptive, secretory in function, and there is an addition of goblet cells in the simple columnar epithelium, because we need mucus also in the small intestine as well as the large intestine. So, in the large intestine, again we have the simple columnar epithelium, which is absorptive, secretory, and goblet cells are present in large numbers. Why? As we go down the large intestine, the number of goblet cell increases. It is in the rectum, it is in ratio of 1 to 1, one colonocyte and one goblet cell. Why is that? Because again the waste product is becoming hard in the form of feces. Water is getting absorbed and the waste product is getting harder. So, we need excessive lubrication in the lower part of the large intestine, so that the feces can be passed out easily. Okay. So, lamina propria, as I have told you, the epithelium is always associated with an underlying layer of loose connective tissue, and the layer of the lamina propria, it varies in thickness in various parts of the GI tract, and it contains the apart from the loose connective tissue components, the blood vessels which supply the blood to the avascular overlying epithelium, and the lymph vessels, the lymphatic channels, lymphatic capillaries which collect the lymph, as well as it absorbs the fat in the small intestine.
It has got very important function in the small intestine, that the fat gets absorbed in the lymphatic channels, not in the blood vessels. Then the nerves, and in some places there are glands in the lamina propria which are serving the function of secretion of different substances, enzymes as well as the juices. Now, mucosa associated lymphoid tissue. So, our mucosa is exposed to the food particles which we are eating, variety of food from hygienic or unhygienic. So, immunity is provided in the layer of the first layer of the GI tract, first layer of the wall of GI tract, so that any bacteria or microorganism getting entry from this route can be killed. So, mucosa, in the mucosal lining in the lamina propria, there is lymphoid tissue. Now, this lymphoid tissue can be in the form of aggregates. You can see here excessive amount of lymphoid tissue. You can well appreciate here, this is the ileum and this is the appendix. So, you can see in the lamina propria the excessive amount of the lymphoid tissue. It acts as immunogenic barrier against the ingested antigen. Now, this is the colon and this is the jejunum. So, you can see here the isolated lymphoid tissue. So, malt can be in the form of aggregates of lymphoid tissue or the isolated lymphoid tissue, lymphatic nodules, isolated lymphatic nodules. So, the previous one was the aggregates of lymphatic nodules, while this is the isolated lymphatic nodules which are lying in the lamina propria of the tract of some part of tract of GI. Now, lymphatic nodules, it is very important to draw lymphatic nodule in your practical copies, in your exams, and the understanding of it is very important for you, because the examiner often asks question about it. So, here I would like to emphasize on how you would draw the lymphatic nodule. First of all, what is a lymphatic nodule? It is an aggregate of, so students usually they don't know about the lymphatic nodule. What is it? So, there is a difference between the lymphatic nodule and the lymph node. Lymph node is the organ in which the lymphatic nodules are present. This is the lymph node and these are the lymphatic nodules which are present in the lymph nodes also. So, lymph node is an aggregate of what? Aggregates of lymphocytes. Now, there are two types of lymphatic nodules. This one shown here has got darker shade in the peripheral region, while the central region has got lighter shade. So, this is the secondary lymphatic nodule. This is the secondary lymphatic nodule, showing the germinal center, lighter central zone, which is the germinal center. Okay. Now, how to draw it? So, first of all, before drawing this lymphatic nodule, you should have the knowledge of the lymphocyte. Lymphocyte, how you will draw lymphocyte? Lymphocyte is drawn like that, large nucleus with rim of cytoplasm around it. So, it has got a large nucleus with very little cytoplasm. This is the lymphocyte. lymphocyte. Now, how you draw a lymphoblast? So, in the germinal center, the cells are lymphoblast, while in the periphery, the cells are lymphocyte. So, lymphoblast has got a larger size of the cell and a smaller nucleus with excessive amount of cytoplasm. So, they give less staining. So, they take less staining. So, they give less appearance of the staining, that is the hematoxylin, while the peripheral cells are the lymphocytes, which have got a large nucleus, and they give more color, hematoxylin, purple color to the region. So that's why the periphery appears dark, while the center of this lymphatic nodule appears lighter due to presence of the lymphoblast. So next layer is the Muscularis mucosae. It forms the boundary between the mucosa and submucosa. This always consists of thin layer of smooth muscle, and this can be arranged into two layers, concentric layers, inner circular and outer longitudinal. So now here in this is the histological section of esophagus, and you can see here the epithelium. This is the epithelium, stratified squamous non-keratinized epithelium. The next layer is the lamina propria, and this layer is the muscularis mucosae layer. You can well observe here the thinness of the layer as compared to the second layer, that is the muscularis externa. How much it is thick, and how much the muscularis mucosae layer is thin. So it is usually arranged in two layers also, inner circular and outer longitudinal. It causes local independent movement and folding of mucosa that facilitate digestion and absorption and increase the contact of the food with the surface epithelium. So to promote digestion and absorption. So next layer is the submucosa. It also consists of the loose connective tissue, but it is rather dense, somewhat dense as compared to the lamina propria. Now here you can see the mucosal layer. The three parts of the mucosa is the lining epithelium, then the lamina propria, and then this thin layer is the muscularis mucosae. Next is the submucosa, and next to submucosa is the muscularis externa, and the last layer is Sirosa or adventitia. So the submucosa, it lies between the mucosa and the muscularis externa, and it contains relatively large blood vessels as compared to lamina propria, also the lymphatic vessels and the nerves, and a special nerve plexus. No glands are present in the submucosa except in two regions of the GI tract, that is the esophagus and the duodenum. So in esophagus and duodenum, special mucus glands are present. For the secretion of excessive amount of mucus. Okay, so submucosal nerve plexus, also known as Meissner's plexus. It is a network of neurons and interconnected unmyelinated nerve fibers present in the connective tissue of submucosa. So most of the nerve cells are motor neurons, but sensory neurons and interneurons are also present, and they are the part of enteric nervous system, and they innervate the muscles of muscularis mucosae and the cells of the intestinal glands, as well as the glands present in the lamina propria. Next layer, muscularis externa. This is two cell thick concentric layers of smooth muscles, not always smooth muscles. In some region, few region of the GI tract, skeletal muscles can be present in the muscularis externa. We will see when we will study the individual region. They are usually arranged in inner circular and outer longitudinal layer, usually two layer thick, and their contraction mix and propel the contents of the digestive tract, and they produce rhythmic contractions of the muscle layer of the muscularis externa, producing waves of contraction in the wall of digestive tube, which is known as the peristalsis. Now here you can see the muscularis externa layer. It is the inner circular and outer longitudinal. So this is the circular layer arranged concentrically around the lumen, and this layer is running longitudinally like this. So when we cut it, the cut section appears like this. So muscularis externa of stomach. At some locations along the digestive tube, the basic muscle pattern in muscularis externa is modified by localized increase in the circular layer of smooth muscle, such as thickening of the circular layer of smooth muscle produces the sphincter in the pyloric region, while also the wall of the muscularis externa of the stomach contains 3 layers, not 2: inner oblique, middle circular, and outer longitudinal. Because stomach has to perform powerful churning movements, so there are 3 layers in the muscularis externa of the stomach. Now this is the colon, and you can see here the inner circular layer is good, but the outer longitudinal layer is arranged in the form of bands. It is not encircling whole the organ, it is present in the form of bands. These bands are known as the tinea coli. So we will study them when we will study the colon, but here I want to show that the muscularis externa can be arranged in 2 layers: inner circular, outer longitudinal. Can be arranged in 3 layers: inner oblique, middle circular, outer longitudinal, and the outer longitudinal layer can be present in the form of bands called the tinea coli in case of colon. Okay, so important nerve plexus, myenteric nerve plexus, which is also the component of enteric nervous system, is also present between the 2 layers of the muscularis externa. Between the layers of muscularis externa, they contain motor neurons, sensory neurons, and interneurons connected to each other by unmyelinated fibers, and they innervate the smooth muscle of the muscularis externa, and they are responsible for the production of the peristaltic wave. Okay, last layer is the Sirosa or adventitia. Now this is very interesting. The last layer of the organ consists of the basically the connective tissue, which is adhering it with the other structures. But in case of GIT, as in case of lungs or heart, these structures are moving. When they move, they produce friction with each other. So to minimize this friction, the organs of the GI tract, the tubes of the GI tract, they are covered by peritoneum, 2 layers of peritoneum, that is the visceral and parietal, and between these 2 layers is present a thin film of fluid which prevents the friction. So whenever the digestive tube is covered by the peritoneum, visceral peritoneum, completely surrounded by the visceral peritoneum, this is known as the Sirosa. And histologically, there is a layer of simple squamous epithelium added to in addition to a loose connective tissue. So the adventitia consists of the loose connective tissue, and in this loose connective tissue lies the blood vessels, nerves, lymphatics, which are entering or leaving the organ, and the some amount of adipose tissue also. And in case of Sirosa, a layer of simple squamous epithelium is added. So this is all about the general histological arrangement of the layer of GI tract, wall of the GI tract.